Studies that are in vivo are those in which the effects of various biological entities are tested on whole, living organisms, usually animals including humans, and plants, as opposed to a partial or dead organism, or those done in vitro, i.e., in a laboratory environment using test tubes, petri dishes, etc. Examples of investigations in vivo include the pathogenesis of disease by comparing the effects of bacterial infection with the effects of purified bacterial toxins, the development of antibiotics, antiviral drugs, and new drugs generally, and new surgical procedures. Consequently, animal testing and clinical trials are major elements of in vivo research. In vivo testing is often employed over in vitro because it is better suited for observing the overall effects of an experiment on a living subject. In drug discovery, for example, verification of efficacy in vivo is crucial, because in vitro assays can sometimes yield misleading results with drug candidate molecules that are irrelevant in vivo. The English microbiologist Professor Harry Smith and his colleagues in the mid-1950s showed the importance of in vivo studies. They found that sterile filtrates of serum from animals infected with Bacillus anthracis were lethal for other animals, whereas extracts of culture fluid from the same organism grown in vitro were not. This discovery of anthrax toxin through the use of in vivo experiments had a major impact on studies of the pathogenesis of infectious disease. The maxim in vivo veritas is used to describe this type of testing and is a play on in vino veritas, a well-known proverb. In vivo versus ex vivo research. In microbiology, in vivo is often used to refer to experimentation done in live isolated cells rather than in a whole organism, for example, cultured cells derived from biopsies. In this situation, the more specific term is ex vivo. Once cells are disrupted and individual parts are tested or analyzed, this is known as in vitro. Methods of use. According to Christopher Lipinski and Andrew Hopkins, whether the aim is to discover drugs or to gain knowledge of biological systems, the nature and properties of a chemical tool cannot be considered independently of the system it is to be tested in. Compounds that bind to isolated recombinant proteins are one thing, chemical tools that can perturb cell function another, and pharmacological agents that can be tolerated by a live organism and perturb its systems are yet another.